Hi everyone, today we are going to be talking about prokaryotic replication fork. So here listed are just a few things we're going to be talking about today. But first off, what is the function of the DNA replication fork? And the function of a DNA replication fork is the area where replication of DNA takes place. We have to start to look at this at the origin of replication. Now, this is different in prokaryotic and eukaryotic organisms, but both is the place where replication occurs. Drawn is the origin of replication for prokaryotic organisms, which is circular DNA with one replication bubble, but eukaryotic is linear DNA with multiple replication bubbles. So after we have the origin of replication, helicase comes in, and helicase unwinds the DNA and unzips the genes. After helicase unzips the genes, single-stranded binding proteins come out and coat the single strands, and this prevents the single-stranded DNA from winding back up into the double helix, because that would be bad and replication couldn't occur if it went back up. And then gyrase also comes in with single binding proteins and prevents the DNA from being over or underwound. So you can see that drawn in pink. Then we have RNA primase that comes in after the gyrase, and the RNA primase synthesize RNA primers to allow for DNA synthesis to begin. So finally, DNA synthesis is about to begin, and it actually begins and synthesizes DNA with DNA polymerase III. So DNA polymerase III comes in and synthesizes the DNA, beginning with the free 3' hydroxyl group. You can see that written in that picture. And then we have leading versus lagging strands, which are important. They are two strands of DNA that are anti-parallel to each other, and the leading strand and the lagging strand both run 5' to 3', but the lagging strand loops around the replication fork, so it all proceeds in one direction. Replication happens continuously on one strand, the leading strand, and discontinuously on the lagging strand. So after looking at leading and lagging strands, we can talk about Okazaki fragments. And these are short links of DNA produced by discontinuous replication of the lagging strand. So Okazaki fragments are only on the lagging strand, and you can see that written in red in the picture. And then DNA polymerase I comes in, and it cuts out the RNA primer, and it pretty much removes the primer and fills the gap. Now, what's the importance of 5' versus 3', and what does this really even mean? So it pretty much just gives a DNA a direction. 5' carbon has a phosphate group attached, and a 3' carbon has a hydroxyl group attached, and it gives DNA strand a direction, either 5' or 3'. And then last but not least, we have DNA ligase. And ligase comes in and pretty much seals the nick. It seals the DNA, which creates phosphodiester bonds, and phosphodiester bonds are linkage between a 3' carbon atom of a one sugar molecule and a 5' carbon atom of another. And that is pretty much the end of my presentation. That is summed up the prokaryotic replication fork. Thank you very much.